Great. Hi, everyone. My name is Dr. Lisa Robeson, and I'm currently a visiting assistant professor at Stetson University. I'd like to really first thank Open Box Science for the fantastic job that they've been doing putting together this series. I think it's really helped me and probably a lot of other people come together, especially in the face of COVID, to just meet a lot of fantastic people, hear about some of the great work that's been done. And especially a big thank you to Dr. Jerry Lynn for the opportunity to moderate today's session. So just a quick update on today's schedule. So unfortunately, Shima Safayan, who is scheduled to present her neuron paper on white matter aging drives microglial diversity, she won't be able to join us today, but we're hoping to have her rescheduled for a session later on in July so you can hear about some of her really fantastic work. But we do have with us today Dr. Nicola Fattorelli, and he'll be presenting his paper, Stem-cell-derived human microglia transplanted into mouse brain to study human disease, published in Nature Protocols earlier this year. So a little bit about Nico. So he's an Italian graduate student, and he received a bachelor's in biotechnology and a master's in neurobiology from the University of Trento, Italy. And since 2018, he's been in the lab of Bart de Struber at the Flanders Institute for Biotechnology in Belgium for his PhD to study the role of microglia in Alzheimer's disease. So a few fun facts about Nico. So he has two of his favorite scientists that show his Italian pride, and that is Leonardo da Vinci and Galileo. And of course, he's also a huge fan of Pio del Rio Ortega, who discovered microglia, who he says are the best cells ever. Another fun fact that I'm definitely asking about in the Q&A session is that there's a legend that he had a goat joining one of his lab meetings. So I don't know how exactly that was involved, but again, we'll hear about that later. So Nico, if you want to take it away, we're very excited to hear about this protocol that you came up with.
Thanks a lot for the very kind introduction. I'll start sharing my screen. And in the meanwhile, I also wanted to thank you guys a lot for the invitation. That's really a great opportunity. I think that you give to trainees to be able to present their work. So yeah, it's great to be here. So today, as Lisa introduced, I'll be presenting our microglia xenograft model that we developed in the lab. And I'll start from Alzheimer's disease because that's what we are mainly interested in the lab of Bart de Struber when I'm doing my PhD. But then I'll try to convince you that this model is also great to study a number of aspects of human microglia physiology and disease. So as I said, I like always to start from Alzheimer's disease because you know that among people over 65 years old, 10% of them will eventually develop AD. And the vast majority of these are sporadic cases. But studies in twins actually indicated that 60 to 80% of the risk to develop AD in these sporadic cases is actually still genetic. And dozens of genetic risk loci have been recently associated to AD by genome-wide association studies in large cohorts of patients. And you see here an example of a Manhattan plot where basically the work of geneticists is trying to associate these SNPs to genes and to link these alleles to either a risk or a protective role for AD, respectively genes depicted in blue and in yellow in this view. And an interesting observation that we made a couple of years ago in the lab, looking at our and others' work, is that especially from single-cell datasets, we could observe that up to 90% of the risk genes associated with AD are actually expressed in microglia, which you know are the resident immune cells of the brain. And many of these genes are also specific for this cell type in the brain. And from a functional perspective, if we do data mining of this list of genes, this shows that they're involved only in a limited number of pathways. And on one side, we find the immune response, of course, where, for example, TREM2, CD33, or HLA were found. Or on the other side, another main pathway that is identified is lipid or endocytosis metabolism, which include, for example, lipid transport through apolipoproteins, lipid receptors, and a number of genes which are involved in endocytosis mechanisms. Now, very interestingly, what we recently observed is that up to 50% of these risk genes may be difficult to study in mouse microglia models. And this is because these genes are either not expressed in mouse microglia, or they don't have a clear one-to-one ortholog between human and mouse. This observation was also confirmed by other studies, and one that I like to cite is a large study by Ido Amit and Marco Prinz labs, who really showed in Cell a couple of years ago the unique heterogeneity of human microglia compared to other species, and even compared to other primates, and especially how these human microglia expresses specific genes which are relevant to neurodegeneration. So that's why we decided to. My slides are blocked, sorry. They don't go. Okay, cool. Sorry. This is why I was saying we set out to develop a new system to study genetic risk in human microglia. And what we did, we started by differentiating pluripotent stem cells towards microglia progenitors in vitro. But microglia, when plated in a dish, they commonly displayed this artificial phenotype that you can see in the picture, and they don't really look like microglia when they are in vitro. That's why we developed this model of transplantation into the brain of immunodeficient mice. And this is how the same cells look when transplanted in vivo. So you see that they completely change. They look beautiful. They show this typical morphology, ramified of microglia cells. Their typical mosaic distribution in the brain. They show expression of human microglia markers. And in our first proof of concept work that we published in Nature Neuroscience, we show how in our chimeric model, the transplanted cells could integrate and survive in the mouse brain. And that this in vivo environment was sufficient for the cells to become human microglia. And how we did that was by comparing microglia progenitors in vitro to our xenotransplanted cells, and 8 weeks after injection, and to primary microglia that we were able to access from healthy tissue removed in surgical resections from the neurosurgery here at KU-Leuven. And we performed tissue dissociation of these brains and sorted human microglia and did single-cell RNA sequencing. So the clustering analysis showed how this transplanted microglia almost completely recapitulated the transcriptional signature of the primary cells. You see that they group together in this cluster that we call the in vivo homeostatic microglia, which is driven indeed by high levels of homeostatic microglia markers, and with almost no overlap with the cells that were coming from the in vitro cultures. And if we look at the homeostatic markers in the heat map on the right, you can see that it's clear how the transplanted and the primary cells express much higher levels than the in vitro counterparts. And vice versa, we observe for the activation markers. So, in fact, when we compared by differential expression the primary microglia with the in vitro cells, we found many differentially expressed genes which were pointing again at an overactivation of the cells in vitro, while when we compared the primary cells to the xenotransplanted ones, these differences almost disappeared. So to test if our transplanted cells were able to respond to challenges, what we did was to perform an acute injection of oligomeric A beta for 6 hours in mice aged between 8 to 10 weeks. And again, we dissociated the tissue, and this time we sorted both human and mouse microglia. And we compared the response to this AD-related challenge of oligomeric A beta injection by single-cell RNA sequencing again. So as you can see from this correlation plot, while the gene expression changes upon A beta treatment positively correlated between the two species, you have human on the y-axis and mouse on the x-axis. The cytokine response to oligomers, which you see is characterized by L1 beta, CCL2, CCL4 upregulation, was more pronounced in human microglia. And you can see this particularly from this bar plot, where I'm highlighting the top and the bottom upregulated and downregulated genes upon oligomeric A beta treatment in the two species. And you see that the blue bars for human cells are, the reaction is more exacerbated. But interestingly, what we also observed is that there was a set of genes which were reacting in opposite directions if we were comparing the two species. Again, showing species-specific differences besides the overall correlation. And if we focused on Alzheimer's disease risk genes, coming back to my introduction, while some changes you see that went in the same direction upon A beta treatment, there were a number of genes that significantly changed only in human microglia. And one example can be SOREL1. And some responses, which I'm highlighting in green, were actually going in opposite direction. And this is, for example, the case of BIN1 and PCAN. So coming back to those risk genes, we tried to put together a bit the evidence that we and others collected and about how they compare in human versus mouse microglia. And here I'm just highlighting a subset of them. You'll find the full figure in the paper. But as you can see, some of them are already difficult to model in mouse microglia in principle because orthology is not clear. We don't have a one-to-one clean ortholog. And for some of them, the sequence is also divergent from human, or as we discussed before, some of them are only expressed by human microglia. And we checked this in a number of datasets, including this one that I just showed and other published mouse datasets. So with this, in this study, we label some of these genes as potentially problematic, so difficult to study in mouse. And we've just seen actually that even those that are expressed by mouse can even react differently if compared to human in an AD-related challenge. So with this first study, what we show is that our chimeric models are a great tool to fill this gap. And they allow investigation of human microglia genetics in brain disorders. And since we were convinced that the potential of this model could go beyond that, we wanted to get it as efficient and as consistent as possible. So our next question basically became, how can we get better chimeras? Because if I show you the grafting efficiency of the human microglia into the mouse brains of our proof of concept study, you see that while we were able to provide evidence for the survival of these cells, as I've shown you before as well, the engraftment efficiency, which we define as the proportion of human microglia out of the total pool of microglia in these brains, wasn't overwhelming. You see it's quantified in the green bar, and it was around 10% on average. So we decided that we wanted to invest efforts in the technical advancement of our protocol. And that's why we started this project, which I shared with Ana Martinez in our lab. And to do so, we went back a bit to the literature of microglia differentiation protocols. And we saw that this is actually very, very crowded. There are many, many protocols from a number of different labs, which are all similar to each other, but they all differ from for the cytokines that are used, the time points, and so on. But our intent wasn't to throw yet one more protocol on this pile, but rather we wanted to refine the existing ones, and in particular the one that we used in our study, to maximize this grafting efficiency in vivo that I was just showing. So we took this grafting efficiency as a readout. We tried a number of protocols. Some of them worked, some didn't work when we grafted cells. And it was by playing with the cytokines and the time points of the differentiation that we found an interesting candidate that we named migrate. And we followed it up and we characterized thoroughly. So basically, this was by introducing some modifications to the in vitro procedure of our original study. And but we were really able to optimize substantially the xenotransplantations. The paper is recently published in Nature Protocols, as Lisa was mentioning, and what we show basically is a consistent and very efficient transplantation across a number of different embryonic stem cells and iPSC lines, which already after 3 weeks after injection, we find a transplantation efficiency that reaches an average of 30% across different lines, and which increases up to 70–80% already after 2–3 months after transplantation. So you can appreciate this also in this sagittal section from a transplanted mouse, where every green ramified cell that you see is a human microglia, positive for human P2R-Y12. And you realize that we name our protocol migrate because while we graft the cells into the striatum of these mice, they're really able to proliferate and to migrate across the full sagittal section, as you've seen, across the full cortex, to the hippocampus, and even reaching the more caudal regions of the cerebellum. So now, having such a model available, we can really start asking mid or high throughput questions in the in vivo environment, which is really, really exciting for us. And here is just to say that in the paper we describe the procedure really step by step in detail with all our tips and tricks that we found during the optimization of the protocol. And yeah, we're really happy if you guys ever want to try it. And of course, don't hesitate to reach us for questions. Of course, we are currently exploring a number of research lines in the lab using the migrate protocol, from physiology of microglia to development and genetics of disease, of course. And yeah, we're really curious to see what will come up, and also from you if you'll ever want to try it. So yeah, I want to thank you for all your attention, but first, of course, all of this wouldn't be possible without the help of many, many people. I'd like to thank Bart, of course, for supervising me, but also many other people in the lab, including Lane and the microglia team, but especially Renzo Mancuso, who pioneered the work on microglia chimeras in the lab. I learned a lot from him, and he's now an independent group leader at VIB Antwerp. And Anna, who shared with me the work on the migrate protocol, as I mentioned. Lastly, the funding sources, especially FWO, for funding my PhD. And thank you all for joining and for the attention. And I'm happy to take any questions that you should have.
Thank you so much, Nico. That was really amazing work. I mean, I'm just continuously in awe at kind of the speed at which all these new methodologies are helping us drive the field forward. And, you know, I'm just incredibly hopeful for the future of us being able to find better treatments and cures, hopefully, for some of these diseases. I know you mostly focused on Alzheimer's disease here, but in your paper, you talked about other diseases like Parkinson's and schizophrenia, as well as things like viruses like Zika and HIV. So great work, really, really exciting. So if you have any questions, you can start putting them in the chat. We have one right now, but we'll be happy to take 20 more. Okay, so we have our first question from Eduardo Macora. And he said, with a few exceptions, the AD risk genes that were shown are just labels for genomic regions that contain several genes. They have not been validated as AD risk genes. Have you looked at the response of all genes within or in the vicinity of these regions to gain information that could be used to map the causal genes?
Yeah, that's a great point. I know Eduardo is a geneticist, so yeah, absolutely, I agree. We haven't looked at all genes within the vicinity of these regions. That's for sure something that we can do. And yeah, as I mentioned in the beginning, the work of geneticists is very, very important, and all these genes need to be validated. SNPs need to be assigned to their correct genes, and for sure it's a very interesting exercise to look at all these genes that are covered by the region of these SNPs. We haven't done that yet.
So we have a few questions from Luca DeFeo. I'm going to just give you them one at a time because they have three. So first they said, nice talk, but just three questions. So the first one is, did you also check to see if the transplanted cells are migrating into the spinal cord?
Oh, no, we haven't checked that, but we can do it.
Okay. Second question is, what is happening to the endogenous mice microglia?
Oh, yeah, that's also a good point. I forgot to mention maybe that before transplanting the human cells, we do deplete for the mouse endogenous microglia. We do a 2 days treatment when the mice are postnatal day 2 and day 3 with a selective inhibitor of CSF1R. So we deplete because we want to give a permissive environment for the human microglia to engraft and occupy their niche. But of course, we know from several studies in mouse that once endogenous microglia are depleted, they will start to repopulate very fast. So the moment we engraft the human cells, basically there's a competition that starts between the two species. And what we saw is that human cells do have an advantage in this competition, and they are more able to take over and to occupy the niche. And even though we will still have always a bit of mouse cells, with time, as I saw with these bar plots of grafting efficiency that we checked at different time points, with time we see that the human cells really occupy more and more of their niche. But even though we never observed overgrowth, we never observed tumor-like structures, they always maintain this mosaic-like distribution, the human cells. And maybe another interesting aspect is that we observe that the human and the mouse cells, they always occupy regionally different areas. So they never intermingle in the same area of the brain, but it's rather that the border of the regions of human and microglia, mouse microglia are touching, and then with time the human are slowly taking over without ever overgrowing. So that's a really interesting point of microglia biology that we can look at. Yeah.
Question number 3. Did you try to transplant any of them into the CSF and not into the parenchyma?
No, we only tried the parenchyma. We inject in the striatum. And yeah, as I showed in the images, but we never tried CSF.
Okay.
Could be interesting.
And just kidding, they actually had a fourth question, sorry. So could we say that we cannot differentiate functional microglia in vitro because their immune phenotype is tissue dependent? So potentially all the macrophage progenitor-like cells in vitro might become microglia when transplanted in vivo.
Yeah, that's also an interesting point. I would tend to agree because on one side we see that the in vivo environment is really the key factor here, right, for these cells, these progenitors to become microglia. But on the other side, I briefly showed a couple of protocols that we tried to transplant, and not all of them worked. For some reason that we cannot explain, some of these protocols were not able to engraft. Some, like the protocol that we use in Nature Neuroscience, had a decent but not too exciting grafting efficiency. And then we eventually came up with this final migrate protocol that is really good at grafting. Now, we don't know yet why. But what I can speculate is that probably with the specific combination of cytokines and the time point where we collect the cells and we graft them, we are transplanting indeed, as the question was mentioning, a particular state of macrophage-like, microglia-like progenitors, which really hold the key for the colonization of the brain. And maybe in other protocols at other stages are not able to do. So yeah, I partially agree, but I also think that the in vitro protocol is very important.
We have a question from Jerry Lynn. Do you see any changes in astrocytes after transplanting the human microglia?
We also haven't checked that yet. So far, we are focusing on the cell autonomous aspects of these transplantations. We really have a lot of aspects of microglia biology which we want to look at. But definitely the next step is looking at interaction with other cells. Of course, in this case, it will be mouse astrocytes or mouse neurons if you would look at interaction. But still definitely an interesting aspect that we could look at with this model, especially with this kind of engrafted efficiency where we find cells in all regions of the brain. So one could really go and look at specific interactions.
Great. So I have a question. So with this updated protocol, what is like the latest time point after transplantation that you have checked to see this chimerism persist?
I think it's by now 18 months that we checked. So 1 year and a half. And we have grafting efficiencies very, very high, up to almost 100% of the
So it
brain.
maintains
Yeah.
after. Okay, great. So,
but we never see overgrowth. That's nice, actually, as I was mentioning before.
Great. And I have personally one more question. So I'm a sex differences researcher, so I was bringing this up. And I know in the original paper you mentioned, where the authors mentioned using mixed sex mice. And I was wondering if there had been any kind of sex-specific analysis to look at efficiency or differences in morphology, since we do know there are sex differences in microglia and risk for some of these kind of microglia dependent diseases.
Absolutely. We haven't looked with a specific protocol at sex differences. So on one side, we tried indeed to use iPSCs and those embryonic stem cells are coming both from female and male patients, actually. But we haven't looked at gender differences yet. But definitely we know that there are gender differences in the microglia reaction. And actually, a work from our lab of 2 years ago, looking at the reaction of microglia in APP and LGF with single-cell RNA-seq, we also really were able to find that the female cells in that case, the female microglia in female mice were reacting much faster than the male ones. So it's an interesting point also.
Yeah. So I think that's all the questions in the chat. I have two kind of fun questions. So I love being able to talk to the author and kind of hear the background of the science. So do you have like a favorite moment or like that Eureka moment that was really memorable that stuck out in your head where you're like, yes, this is actually going to work? Anything like that?
That's a good question. Maybe, yeah. I will cite the moment where, this took a lot of optimization. We really tried a lot of protocols and we went through literature. We tried different combination of cytokines, time points, different collections. So this was really a lot of work because we really wanted like a nice protocol that was consistent and that we could share with the community as well. So I remember the moment at the FACS facility when doing all this grafting efficiency. We were doing it every week and grafting every week, all these protocols. Once we saw this combination that we ended up calling migrate, really working much better than the others that we were trying. That was really a nice, maybe not Eureka, but a really nice moment that which followed with a lot of nice details.
Great. So we did have a few more questions come in while you're giving us that little background. So Mohi Rostagi said, nice presentation. Did you see microglia activation either in the human-derived or the residing mice after transplantation? So either in the human or the endogenous mouse microglia cells.
I guess I don't know if activation here she means like an inflammation-like reaction due to the injection, or if they rather mean activation due to disease. So in case it's the first. It's the first case. Yeah, well, you do see at the site of injection in the first days, weeks that there is inflammation occurring, of course, but then in the long run that you don't observe anymore. But if this was a disease-related question, well, we are looking into that. We have a number of projects running in the lab, working with AD mice, and how these inflammatory reactions of microglia look like across time, and genetics. We're looking at different genes, and yeah, we see a lot going on, but it's all preliminary, and it hopefully will come out soon. But we do see reaction. We're looking at human, and the question was asking at both species. In this proof of concept, we compared human versus mouse. Because yeah, in our first study that was of course interesting to see, but now we are mostly looking at human.
Great. And then Eduardo has another question. So, but what do you make of the fact that most studies that looked at human microglia and human AD brain did not recapitulate the microglia response that's observed in mice, while you observed a pretty good correlation of the mouse versus human brain environment?
Yeah. Yeah, that's a great question. It's also a big point of discussion in the field, definitely. On one side, I think it's probably technical, because looking at post-mortem samples, we know that the samples themselves have limitations. And also with post-mortem samples, you can only work with the single nuclei RNA-seq, and there was actually a paper from our lab last year showing that single nuclei, at least with the technology that was available up to a couple of years ago when we did experiments, single nuclei was not able to recapitulate technically the full activated microglia signature. So it may be also for a technical reason. We had a side-by-side comparison with some of these surgical resection samples that I also showed before in the presentation, doing single cell and single nuclei from the same samples. And in our hands, and then also looking at some of the big published data sets of single nuclei, we saw that the technique itself wasn't powerful enough. Now, on one side, I think that this is improving. I think that some of the latest papers, I remember the Bodicki lab paper that came out some months ago, describing these AD1 and AD2 clusters, is already finding more and more genes related to the signature. So I'm confident that technology is improving, and probably we can find the same back. But then for the second part of the question, how do you explain the correlation that we see? Well, on one side, I guess it's, as I already replied, because we do single cells, so I suppose that this is more sensitive and we can pick it up. But yeah, this is also, I wanted to comment, is probably a reassuring evidence for us and for our model, because if we can overcome the limitations of post-mortem samples and the fact that you need to use single nuclei with a xenotransplanted model where you have human microglia in an in vivo environment, which actually shows this phenotype, it's also a nice confirmation that what was seen in mouse was true. And we can now start to look into the much more heterogeneous reaction that the human microglia have, as I was mentioning in the beginning, that is shown also by Domit, Marco Prinz, and others.
Thank you. So I think that brings us to our last question, unless anybody else has any other questions, comments. Okay, great. Well, thank you so much. It was really fantastic work and beautiful presentation. And I really admire your persistence, because I know protocol optimization can be kind of a heartbreaker, but I'm glad it all worked out for you. Congrats.
Thanks a lot, Lisa, and the guys of OpenBox Science. This was a really great opportunity for me to show our work. And yeah, see you next time, I guess.